In the standard protocol, different samples are sequenced in different lanes of the Illumina flow cell. And that makes it more expensive because you have to pay per lane in Illumina sequencing. And it also introduces noise because different lanes, can be a bit different with respect to sequencing efficiency and so on. To reduce sequencing costs and to avoid introducing noise in the data, samples can be sequenced in the same lane of the flow cell. This is only possible when there is a way to find out for every sequence that is generated which sample it came from. And this is where the antibodies come in. So antibodies are used that target a very general cell surface protein that is on the surface of almost any cell. And they are linked to a piece of DNA similar to what you see here with a poly-A tail, with a part that will bind to sequencing primer and primers that are used for PCR. And instead of a unique barcode that is specific for every antibody, we will use a sequence that is specific for every sample. So what you will get in this system is here a set of antibodies that target very general cell surface proteins. They are all linked to the same sequence that is specific for sample number 1. You have another set of the same antibodies, but this time they are linked to a sequence that is specific for sample number 2. Each set of antibodies is allowed to interact with the cells. You inject your cells into the water droplets, and then using the same system that I explained here, you are going to sequence both the transcripts, so you will get the expression levels of all the genes in every cell, and you are going to sequence the sequences that were linked to the antibodies. And in this way, you will get for every cell the abundance of every sample barcode in that cell. So you can check which sample the cell belonged to. And this process is called cell hashing. So these are not the only two variations on the protocol. There are also protocols available for specifically measuring T-cell receptor and B-cell receptor information, accessibility of DNA, and so on. But we will not talk about these now. There is more information available on the e-learning system for this.